at the end of the room that has a problem.
There is an interviewer bias.
Inter
The question is oriented towards something already.
oh yeah, no, for sure. I'm clear in my question and in my answers which one is the correct out of the three. Oh, here we go. Oop, oop. Okay. So Jaming talked about something really interesting, which was really interesting for me, right? The idea of you have a single space where all the data sits and people connect to it and people can analyze it if you have the right permissions, right? And I don't know if that started or not with that CMG project, the Center for Mendelian Genomics project. At the beginning, every center had their own data. It was complicated to share. And by the end of the project, they decided, okay, let's use the Ambil project. That was the idea of putting everything in one spot. And the only thing you need to do is connect to it and do analysis. The tools will be already there. So you don't have to install anything. Just connect, tools are available, and you do your analysis. But I think the main problem also of that system is that the payment system, you needed to pay. Somebody needs to pay that infrastructure. And it seems it's not that easy. They use one system called Terra, and you can kind of predict how much you're going to spend. But I don't know, how many times do you need to repeat an analysis? 100 sometimes, right? So it's almost impossible to predict how much money you're going to spend. And requesting soft money for this thing seems harder than requesting, oh, I'm going to spend some money in a computing center and do the analysis in my computing center. So it sounds good, but I don't know how easy it is to implement or how many people is actually using it. I don't know, in the case of Korea, I don't know how it's going to work, but the Ambil project, I'm not that convinced. Because the problem is when you create a new barrier, which is the payment system. We have to move all the data that we had in all centers to the Ambil. And from that moment on, I don't know, our centers never use it.
And you lose all your authority over the data then?
If we use
You lose the authority of the data if you put it on the
the quality in the sense,
authority in the sense that you have no hold on that data.
yeah, I mean, that's the thing. It was already public because we have, it was already submitted to NCBI, right, to the SRA, the short read archive. But then we needed to move everything to Ambil to make it centralized. And then we could add more data to that because then you could fine tune the consent, right? So if it was, let's say, only for researching on X, then you can really fine tune who has access to that data. I understand the idea is really good, but I think at the same time you create a new barrier with how you're going to pay for it, I guess. That was my whole point. So I had a few slides to overview. I mean, we have a really good example. You've seen, we've been talking through these days about variant calling, right? There's different talks that talk about it yesterday, today, Jiangming. So I'm going to deal more about the technical part. I probably have way too many slides here, so I'll try to go a little bit fast so we can go directly to hands-on. Do I need to tell you what is variant calling? You probably already know. So it's basically finding the difference with some genomic sequence that you have as a reference, right? You already know this process too. Unlike transcriptomics, now we care about the sequence. Transcriptomics, we only want to align it. Once you align it, you want the count data, right? That's what we know. Now we don't have any count data. What we have is sequence, and we want to see the difference. So the alignment process is really important too, and how we do it. And then we have another process, which is the variant detection. So pretty much we could split it globally in those two steps, sequencing, alignment, and variant calling. We have two big types, or more than two big types, but there are different types of variants. So
So the alignment in transcriptomic data would not be very strict because you just need to
Well, it is strict, right? But it's in the sense that,
For example, here you are trying to call us SNV, so it has to be very
yep.
stricter than, I mean,
And the length of the sequence is more important, definitely, right? So you want the longer the better, so you can cover as much as you can. With transcriptomics, you can work from probably 40 bases up to 150. Maybe for what you want, the result is going to be the same. And here, definitely, you need to pay more attention about sequencing, length, and precision of the alignment, as you say, right? You need.
the alignment algorithms are the same, whether you use
Yeah. I mean, the alignment algorithms are pretty much the same, yeah, or at least are based on the same things, some of them. Oh, yeah, so I was trying to talk that, yeah, so we have different types. We can talk about single nucleotide variants, structural variants, or even copy number variation variants, and combination of those. And Jianming talked about all of them, I think, right? Structural variants in general, even copy number, we put them aside a little bit because you may need different tools for analysis. So we're going to focus on single nucleotide variants today, but anything that's also an artificial definition. But generally we talk everything above 50 nucleotides, so it's affecting 50 nucleotides, we classify them as a structural variants. And also you have different types. Again, today is going to be about single nucleotide variants, not about structural variants, but they exist and they have their specialized tools. Because it's a slightly different problem, right? And that's another interesting thing, which is we are not perfect, right? And surprisingly, we are quite imperfect. There's 3.1 billion bases. We have around 20,000 genes, as we've discussed in these days. The ENCODE project, I don't know how old is this data, but they estimate that 40 to 60% of our genome encodes for something. Generally, on average, right, each person has around that 3.5 million SNVs, 500,000 of small indels. This is another interesting thing. On average, between us, we have around, which we never, or we kind of ignore a lot of times, but among us, so whatever we use as a reference, we assume is the reference, but there are a lot of large rearrangements between us and that reference, or among us. So between 1,500 and 2,500 on average of large rearrangements in our genomes. So when we talk about, generally we say, oh, it's 99.9 equal, the sequence. Yeah, the sequence it is, but we have a lot of large rearrangements that also create some differences. And this is always the part that interests me the most. So we are born already with around, let's say, 300 loss of function variants. Loss of function meaning that gene is coding for something that is wrong. So we already have them. So if we do an analysis, you're going to find variants that they are going to be classified as pathogenic. In general terms, I'm going to call it, but well, better general terms, they're going to be classified as loss of function. So we have them. It can be that it needs to be, that you can have only one copy and the other copy is completely normal, and that's enough. But even if we have a gene that is completely, we have a loss of function of a gene, of a complete gene, the organisms have ways to compensate for that. So, and there are a lot of systems that have been studied, I don't even remember, but all of them, but there are different ways that the body has to compensate. So what I'm trying to say, yeah, so if you try to do an analysis of let's say, a normal person, you're going to find that that person has already 300 loss of function mutations on average. And even what I was trying to explain before, that between even some of those, they're going to be associated to some disorder. So can you give me ideas? You don't have to answer through the computer because we know it's a bit painful. But can you give me ideas why we want to study variants? One of them is rare diseases. We know that, right? Any other? No, you don't have to go to the website.
precision medicine.
Precision medicine? Yeah, pharmacogenomics. right? All related to how we can adapt, or be specific about the treatment.
Organ transplantation.
Organ transplantation, we saw an example, right?
Between genetics, ethnic groups.
Yeah, population genetics.
Association with many diseases.
Yeah, I mean, it doesn't need to be rare diseases. Diseases in general, right? We can study them. It could be a common disease, which is a harder problem. We talk about autism, right?
Ankylosing spondylitis, we know the association
Yep.
with that genetic.
In
Will lead to some disease within too specific.
yeah, I mean, there are good answers. So, I always when I try to think about applications, it always happens the same thing. I put some, and there must be others, and then is when I write a question for the audience, outsourcing to the audience because probably you have better ideas. I think we pretty much mentioned all of them. Oh, cancer research, yeah, for sure, but it's related to any diagnosis, right? Population genetics, agriculture, ancient DNA studies, forensics. Forensics, yeah. So actually, okay, I'm going to play Bonnie's role now. I have a story to tell. So I did ancient DNA long ago. I work in ancient DNA, like 25 years ago or so. And in forensics too, there were a company, I think it was in Netherlands. It was a cosmetic company that was working in shaving brushes. And the high quality shaving brushes, they use badger hair. You know, a badger, it's, yeah. And then it's the species name, there are different ones, but it's Meles, the genus is Meles, something else. So they work with that, and they were worried because they were getting hair from China, or the fur from China, and China, some species are protected. So they were really worried about that. So they sent us the brushes, and we did the study and everything. So the company ended up suing another company in China for sending protected products to the Netherlands. I think it was a year later, CSI, you know CSI, the TV show, the criminal that they do forensics, they did the same thing. So then I discovered the CSI read papers about things, and then they do episodes. So I like to think that they based the episode on our paper. Sorry, what? Oh, no. But I mean, so they changed it. It wasn't a badger. It was a painter, right? A painter that left the hair in somewhere, and it was hair from a badger, and the forensics did the analysis, and they discovered that. Yeah. I think, Bonnie also mentioned this paper. We are proud at the Genomics Center about this paper because it was the first application in whole exome sequencing in clinical settings, right? It wasn't the first paper on whole exome sequencing. I mean, there were 3 papers being worked at the same time. Jay Shenduri was the first one. This was the second one. And then Kaya, which was another investigator. Whole exome, it was Nimblegen, or the first Nimblegen platforms that were available. Or it was a collaboration, I guess, with Nimblegen. And Kaya, which was also at some point work at the Genome Center, was the third paper. Also in a rare disease. Of course, you're going to need to do sampling in your projects, and based on your goal, you're going to do different samplings. Especially, I wanted to mention the difference between, and I'm probably going to mention that later, but if you work with somatic variants, you generally sample the individual twice, the tumor and the normal match, because you're going to have to distinguish what is a germline, what is a somatic. For that, you need both samples. And there are different types of studies, right? You can work with trios, you can work with big cohorts like Jianming's research he showed today. You can work with families or even populations, depending on your goals. Sequencing, we've been talking about a lot of this. Based on the cost, and Jianming mentioned, we can give him different prices. He gives 200, I'm giving 300. I don't know what Bonnie gives, but around those numbers, right? But whole exome sequencing, so this is not necessarily the answer. That's also the point. Because, yeah, you can sequence how many, I don't know the numbers that Jianming gives, but he is going to pay more for storage. He's going to pay more for analysis, and maybe whole exome sequencing is going to be cheaper for all those things and be enough depending on what you're trying to do. Or even you can do targeted sequencing. So actually, when the Center for Medical Genomics started, there were also the Common Disease Project, and they decided to go for genomes, and the Mendelians, we went for exomes. It ended up being more successful, the exome project, than the whole genome project. Autism ended up being more complex than they thought. The power calculations that I have initially about sequencing 100,000 autism cases, they thought it would be enough. Then it wasn't enough, and all of those were whole genomes. Platforms, we already know them. No much secret here. So it's going to be the same type of library preparation. We're going to go through the sequencer, and you're going to get your FASTQ file. So there's no difference from anything we've done so far. You are familiar with the FASTQ file, so I'm not going to go through this. I'm going to stop here a second again, because we talk about the quality scores. I think it was the first day that we were talking about them. We only saw them so far, I think. Well, no, we've done BAM files too. So we see them, a couple of those, right? We saw them for the FASTQC, or the FASTQ files, for the align files. But they do exist also for variant calling. And it's the same type of a scale, right? They tell us how confident we are about the variant call we do, but also for the genotype, meaning is this a homozygous call, is this a heterozygous call? So alignment is a very crucial step. I'm going to just review this again, although we already done it. So we generally work with the FASTA file for our genome, but we need to build an index, right? A lot of aligners work with an index to have quick access to that information. So they don't have to scan the genome every time you need to align or read, right? So all aligners, so if you work with aligners, one of the first steps besides getting the genome is to produce an index of that genome. And every aligner produces a different type of indices. So whatever aligner you're going to choose is going to produce his own index files.
Francis, I have a so if you are doing different kind of sequence, like 150 base pair versus 600 base pair, and you are actually aligning, do all these aligners use different indexing files for that? I mean, like,
Not for the sequencing you do. It's only based on your reference. So the aligner wants an index of your reference. So to quick access it.
no, that I understand.
So he's asking that will the alignment process be different for short read versus the long read? Is that what you want?
Or use of indexing.
So if you have a longer read,
Yeah.
index be longer, larger or something like that?
Well, it's going to depend on the aligner. So we have a specific aligners for long reads, so they're going to produce their own indices. And I don't know, I don't use them often to tell you if there's a big difference in the way they build it.
But the 150 and 600 will not.
Yeah, totally. No, BWA, which is the one we're going to be using, you can align in theory between 70 to 1,000 bases. So in that range, you're good.
What are we doing basically in indexing? What are we doing with our sequence?
So it's getting like a book, right? It's looking for fragments of sequence and creating an index of those fragments. So when you try to access and divide it by chromosomes and all that, so if you're trying to access a sequence, it's going to quickly know what it is in the genome.
So if you want to read one particular line, you don't have to scroll through the whole book.
Yep.
You just look at the index, is page पे इस जगह है, so something like that.
Open that. Whatever.
Because if it does again and again, it's going to take that much longer.
Yep.
Is it?
Yeah. So it seems, I mean, conceptually it seems easy that you say you have a sequence and we're going to be able to align it to a genome. It is not, right? So first, it's very long. Aligners care about speed, basically. That's the most important thing, because if you work with whole genome sequences, it's going to be, you cannot be slow. You need to be fast. So the accuracy, you sacrifice a little bit of accuracy to be more faster. Another thing, our reference genome, or the one that we use the most, it may not be complete, right? I think we talked at some point about those issues.
Is the
Right?
reference genome going to change because there's a lot of talk about pan genome or
Yeah.
like
Yes.
they make different people with different ethnicities
Yep.
and come with a consensus of what people are pan.
That's ideal, but then all the tools are completely different. You throw away all the tools we have for new ones.
How far are we from a pan genome? Like how long will it take?
there are already tools available and you can use them, but as you know, you also mentioned in one of your slides, right? You were talking about most of the clinical data sometimes is AG19, not even 38, right? So databases, everything that is annotated sometimes is from even older versions of the reference, and then you need to transform it to the new one. So we have so much data now in the current reference genomes that it's easier to work with that than trying to do anything else. Right? But I don't know. I mean, they're available. We don't use them, or at least not in our group, right? We don't see anyone requesting them.
It appears that it would be more accurate. It could be more accurate.
That's the idea, yeah, right? That's the idea. So to being able to have all that information in a single reference. Because again, if you compare yourself to the current reference, 300 loss of function variants are going to be there. All the rearrangements, if you can analyze rearrangements, you're going to have thousands of those rearrangements. But that's one reference, right? We know that we are really complex if you look at those numbers.
Or is it possible to make a reference of like we already have like a billion all over the world? We might have a billion so-called healthy individuals.
Yeah, I don't know. I mean, that's a good question. It's something that I never thought.
Reference of that.
Yeah.
It would be easier, right?
Like accessing the variability at once, right? Yeah. I don't know. Yeah, if we can apply that information for alignment and vertical. Yeah. Well, we kind of use it to some extent.
Dr. Franz, the previous slide told that the repetitive regions are masked in the reference genome. So, what do you mean by that?
Sorry, with the AC, I cannot hear you well.
Previous slide.
Oh, the previous slide. Yes. Sorry. Yep.
You told that the reference genome presents complex regions such as repetitive elements, and the repetitive regions are usually masked to avoid them. So,
Yes. Yeah, but I mean, that's a problem you need to face, right, when you align. So not all of them are masked when you align. You mask them later if you want to, but during the alignment, you have what you have as a reference. We don't mask anything. So all the repetitive regions are going to be there, and they're complex. So there's another, right, we have the telomere to telomere genome in the middle before the pan genome, right? But still, we still work with AG38, which is from what, 2013?
So is it so difficult to change everything from there to there?
No, it's not, right? I mean, they are what we call it the shift overs. You shift from one to the other, but you still get weird stuff. I mean, it's not 100% correct. Let's call it like this. You never get a complete shift over. Oh, that's what I was mentioning. Yeah, aligners prefer speed. So they make some assumptions, but the whole idea was that, so aligners need to compromise a little bit to deal with these situations. And another important thing, well, first, one way we're going to deal today is about the idea of soft clipping. So when the aligner maps something and the rest of the gene is not able, the rest of the read is not able to aligner, they soft clip it, meaning it's going to indicate only the part that it's been aligned, and the rest is going to be soft clipped, saying, okay, yes, the read maps here, but there's a section that I don't know where it goes. And the other problem of the current aligners is each read is aligned individually, and that's a problem because they don't align. They're not necessarily is going to be aligned in the same way the next one. So you can be aligning in a region. The first time, the first read aligns in a way. Second time, or another read which is not exactly the same, but they align in the same region, is going to align maybe slightly different. Because based on the algorithm, it thinks that's the best way. But every read is aligned individually. And this is a problem too, or it can be a problem. And I'll give you an example later. Oh, regarding aligners, yeah, again, this is a tiny representation, right? But these are probably popular ones. We're going to use today BWMM. This is a common type of algorithm that I'll mention briefly. Different languages. And we talk about this, being a splice aware or not, right? We talk about HiSat2, which is designed for transcriptomics because it's splice aware. The predecessor was bowtie 2, and then they went to HiSat 2, but bowtie 2 we still use it for some applications. They're based on a slightly different algorithm, but again, they are not that different.
So when we use genomic data, then we use different algorithms.
Yeah, you can use different algorithms. The goal is going to be the same, but you can use different algorithms. Of course, there are aligners like Minimap2, which is designed specifically for long reads. We're talking about pangenome references. So this one is designed for pangenomes. It uses a completely different idea, the graph-based mathematical approaches. Most of those are going to be command line interfaces. They are more flexible and easy to use with big data.
CNC has got a Java interface.
Oh, it has also a Java command line to use it? Okay.
No, the CNC I said has a Java interface.
Oh, it's written in Java, yeah. But there are also some commercial, and I don't know if there's any free ones, but you can use graphical interfaces too for alignment.
Is there any advantage of using the commercial one?
Well, I don't know if I have changed. It's been a long time that I don't look at benchmarks, and I'm blocking the name. Oh, NovoAlign. There's one called NovoAlign, which is commercial. That always shows up as a best aligner. Even better than what we use as standard. But of course, you need to pay for that. So I would say the most common standard nowadays for alignment for genomic analysis is going to be BWA.
NovoAlign was written by Jim.
Sorry, what?
NovoAlign was written by Jim.
No.
What was Jim's alignment?
I don't remember. It was designed for 454 data. This is the basic idea of HiSat 2, also BWA. I don't think it's worth it to go much into this. But the main idea is they take a fragment of your read. It doesn't take the whole read, right? So for a speed and compromise, right, it takes a fragment, looks where can go that read, it maps that, and then tries to extend it. And see, okay, yes, this short fragment goes here. Can I extend the read and keep mapping? Yes, good. Then I'm going to say this is the place. If not, I'm going to look for a different spot. So just to give you an idea, a general idea how it works. They call it a seed. They find a seed and they extend. Seed and extension. The SAM file, we already saw it. I don't know. You have questions about this? But every line in your BAM file represents a read, a read being aligned, and there's information about where it maps. We even look at this is called the flag, or the SAM flag, that it tells us what type of read it is and how it aligns. The position where it aligns. Oh, this is a terrible mapping score. We already know that. This is the cigar string that it tells us how it aligns. It's exactly the same slide, and I remember there was soft clipping. Yeah. So this is the idea of soft clipping. 47 match perfectly. 54 is being soft clipped of that read that doesn't know where it goes. So the first 47 bases matches, the rest of the read doesn't. So I'm going to soft clip it. And it gives me a very low mapping score, so we don't worry about it. SAMTools is the tool for manipulating alignments, and I think we're going to see probably in the Jupyter Notebook again how we can use it. So sequencers are also a problem. They produce errors. PCR produces errors. So the main goal, or one of the goals of the variant callers, or variant calling, is to distinguish errors from non-errors, right? So here I'm just giving an example. So this we're going to be more or less confident that it's a real deletion. Here we're going to be more or less confident that it's a single nucleotide variant. And here we're more or less confident that there are sequencing errors. And there are two reasons. But we're going to go for one. What do you think makes us think, or what is the main reason that makes us think this is an error?
We can't find others like it.
You said because it's at the end? That's an interesting answer, yes. And?
The nucleotide is the same nucleotide is being changed in all reads.
Changing like only C or C.
Yeah, you're going for the second main reason. But yeah, that's another point, right? But there's one basic one. There's even more basic one.
It's not very frequent.
So the reads are not
Very less,
aligned
very less.
sequence.
So yeah, the coverage, right? The coverage that we have of the answer, right? So here we see 3 times, so the deletion and 3 times the reference base. So half and a half, something expected, a heterozygous call. Here we only see it 1 time out of all a few. I know this is very, very shallow sequencing, but if you have more, you can be more confident that this is going to be sequencing error. The other one is the allele. If you analyze the whole allele, you're going to see the C and A. They only appear 1 time when you're expecting the CC combination. Except if it is somehow it has 3 copies, right? We don't know if it has 3 copies, but we can say that. So I'm going to skip this because we already answered. So the main answer is going to be, we're going to base our, or how confident we are about a call is mainly going to be about, the main reason is going to be based on the coverage that we have. The more coverage, the more confident we're going to be about the calls. But that's a good reason too that we're going to go through that because you said it's at the end. We'll talk about that too. I don't need to explain this again, right? We talk about coverage, what it means. So it's how many times we see every base. We also sometimes talk about the breadth. And I think I showed this probably the first day. So I'm not going to stop here. Visualizing, again, the idea of visualizing everything we do is really important. Dr. Amin also mentioned it probably. I don't know if we talk about it, but at least I think it sounds familiar that you mentioned it. IGV as a good tool for visualization. We are not going to use it because it's graphical. I mean, we don't have time for this, but it's easy to install in any laptop and you can visualize your analysis. Not only for variant calling, you can use transcriptomics on this too. You can see how your reads, they splice across exons. That could be a good way to look if you are detecting a transcript with a specific exon or not. You can visualize them too.
You can also visualize splice like variants.
Yeah, exactly. That's what I'm trying to say. The splice variant, right? So you can visualize them using IGV. If you are missing an exon or not in your transcript, it's a good way to do it. Or you mean single nucleotide variant that breaks the splicing? Yeah. That's a good point. Yeah, if you find a variant in the splice site and you want to test later with transcriptomics if that variant breaks or not the splicing, that's another way to do it. Yep. So did I miss something here? Okay, so if you have a call and you don't know if it's real or not, you can visualize it, and you make a good point earlier. So why we think this is a misaligned is because it's at the end of the reads. So aligners have issues at the end of the read. Sometimes, this is what I was trying to say earlier, they're aligned individually. So this, when it was aligned, it was fine. Said, okay, there's nothing here. But then I'm aligning another read that covers the same spot, and I'm saying there is a variant. They are aligned independently, and statistically, this looks like it was the best position for that alignment, and the way to align it was correct. In this case, you need to be careful about variance at the end of the reads, because maybe decides, okay, if I create a gap here or not, it's going to look better. It's most probabilistic thing, right? So it's quite common that, or quite common, if some errors can happen because misalignment at the end of the reads. I'm not going to say it's common, but it can happen. So if the only reason, what I'm trying to say too is, if the only reason that you have to believe that this is real is based on 2 reads and they are at the end, you need to be suspicious. So this is another problem that we know the aligners encounter, which is when you have some repetitive regions, it's going to have a really hard time to decide how it goes, right? So you may create gaps or not gaps, or
This was from Iron Torrent, or this data or from like Illumina?
I don't even remember where I got this image. Oh, I'm telling you, it's from a paper, it seems, of somewhere. Yeah, it's a paper. So now we need to go from this alignments to a list of variants, right? And giving the genotypes for every variant and how confident we are. So that's the main role we're going to do with the variant caller, right? So we go from some alignment to a list of variants. So that's going to be the next step in our process. And also trying to make an effort if we want to classify them, the current models of calling variants. So again, probabilistic method, we're assigning a probability of being, some algorithms, we use an algorithm to assign a probability of a call being correct or not. So these probably are the two popular ones. JTK definitely is one popular one, and it's the one we're going to use, probably the most popular one, but there exist others, and you can even combine them. There's ways that actually our pipelines combine more than one to produce the final list. And of course, we are in the time of machine learning, and they do exist too. Google created its own. I mean, a lot of the different aligners that use machine learning approaches. And
You can give the slide over to him.
to him, no. I'm going to feel bad. And the graph base, what we said before, right? So the pangenomic project is going to we're going to need completely different aligners. Because we don't work with a single reference, we're going to work with multiple references. So that's a completely different paradigm, and we're going to completely need a different type of aligners.
So, but the pangenome will it not make a consensus kind of a thing?
No. It creates a graph too.
So it will align against each of those genomes then?
Kind of. Yes, it creates some sort. There are common regions that are going to have less complexity, other regions are going to have more complexity. So they create some graph approach. Again, I'm not good at it. I never use it. I'm not that familiar. So what about variant callings? You have a long list too. You have a few of them, and they're going to be based depending on your data, right? So we have, depending on what you want to do with data. If you work for single nucleotide variants, you're going to use some types. If you want to work with a structural variants, you're going to use a different type. And if they're short or long read. So they're quite specialized depending on the type of data that you have and your goals.
They're all free to use, or like there are some other like commercial or like
I don't remember which ones they were included here. I took it from another paper. And I don't think this is the complete list. So actually I selected a few because I wanted to give some examples of pangenomics. Which one it is? I don't know if I remember which one it is. Or for example, low frequency. This one is for that in theory was designed to detect very low frequency variants. Like if you have really low somatic variants, or if you work with if you CRISPR screenings, or for CRISPRs, you create some sort of mutation, you know it's going to be in a low frequency. You can use different type of variant callers. Because again, JTK is going to assume that you're working with a human genome, and the expectation is going to be that if he has a heterozygous call, it's going to be 50% one call and 50% the other, right? So some of them make assumptions about the expectations of the data. So that way I don't have to pass every step. The JTK, that's the one we're going to be using. It was created in Broad. It's a command line tool. You need to know that. So we're going to go to the Jupyter notebook. We're going to use it to use command line. So we are not going to work with R today. Well, a little bit, but not much. But basically, it's going to be command line. So if you feel comfortable with the terminal, you can go to the terminal and use it. JTK is a set of tools. That's also important to know. It comes with, it's not just a variant caller. It does other things too. And you can also use it for somatic calling. There's a specialized tool within the toolkit that is designed for somatic calling. This is the website. There's really good documentation in the website. And they have pipelines and what they call the best practices for variant, not the best practices in general for variant calling, but the best practices of JTK variant calling. This is a graph they use to explain the best practices, so all the steps, but I'm going to simplify these in a few global ideas. And I go into the details of 2 or 3 of them to know, but the main goal is going to be to create a file with your list of variants. That's the main goal. We've seen some of this already, but these are going to be the steps I'm going to mention. I'm not going to mention the first one, of course, because you already know FastQC, we already use it. But did you notice something between these 2 steps? We are not trimming anything. We are not going to touch the data, especially today. There are ways you can do some trimming. For that, it's better if you check the JTK documentation. But we are not going to trim anything. We are not going to touch, or we are not going to modify how the read looks like in the sense of quality.
Is it specific when we talk about whole genome, that trimming is always with the transcriptomic data?
No, it's not necessarily only for transcriptomics. There's other applications you could do trimming. For variant calling, generally you're probably not going to do trimming, because trimming is going to modify the quality of your read to some extent. And we want to keep some of those bad quality scores too in the process. So we are not going to touch it. I mean, you could do trimming. So I'm not saying you don't have to do trimming. So in JTK, there's some documentations of ways that you could do trimming to your data. And there's tools for that too.
But if your variant is in the trim,
Yeah, if you remove that section, you may be missing variants too. And ideally, we want to cover as much as we can and not as much as we could. BWA is going to be our aligner. We're going to do one thing that we haven't done for transcriptomics, which is removing PCR duplication. PCR duplication is not giving us more information. It's just adding biases to our estimations, nothing else. So we want to remove it. There are 2 types of PCR. I call it PCR duplication. Let's call it duplication in general, because it can come from the PCR, but it can come also from the sequencing machine. And those are, we call it optical duplications. But there can be a couple of sources where duplication can happen. We're going to do one thing that is called base quality recalibration. And that's why we don't trim. We're going to go back to, we're going to say sequencing machines are giving us not the correct quality scores. We're going to recalibrate them to something that we think is more correct. For the problem of the call is at the end of the read, for example, right? Or because they are homopolymers, let me readjust the alignment. So JTK, not anymore, but I want to talk about this because it's interesting. I think the latest version, definitely, I think from version 4, they don't do realignment or reassembly of your reads. They change the way they do it. But it was an interesting problem to solve, what we said, right? So calls at the end of the read, they may be misaligned, right? You could solve it using what they call local realignment. So I see some variant. Can I get a locally, take into account all the reads, can I locally do a better job that the aligner did?
so local realignment means that it will look at the neighboring read
Yep.
and say that, okay, 10, 20 reads at this are showing this and only 2 are showing, I correct it to that.
Yep. But again, this is not happening anymore. It is not part of the process in the JTK4. That's the one we're going to use today.
So do we get the information in, suppose in 30 reads, these many are of this time band?
Yep, that's part of the BCF format that I'm going to talk in a second. But yes, you're going to get that information. Oh, and of course, we're going to do variant calling and genotype calling in our last step. Or that's, let's say, the GTK alignment process of variant calling. I pretty much mentioned all of them. So I was trying to say that, so when you have some sort of duplication of sequences that you know that are stars and N, they are more or less identical in sequencing, those most probably became part of a PCR duplication. They are not giving you information. They're creating a bias when you try to estimate how many times you see a base, right? So if there is a base here and you're trying to estimate how many times you see, this PCR duplication is creating a bias of the count. So you want to remove them. We don't do this in transcriptomics. It would be great if we could, and probably there are methods that we could do it. PCR duplication can exist in transcriptomics. It can be less if you use less cycles of PCR, but it could exist. But you're working in such a small space, which is the exome or the transcriptome compared with genomics, and duplication is expected because you are sequencing the same transcript multiple times. The optical duplication happens during, Bonnie probably can explain you better. There's some breach process during the sequencing and the spots of the flow cell. And during that process of breaching and duplicating sequence on spot, you create PCR duplicates too. I don't know if there's other reasons for optical duplicates in sequencing.
Yeah, so optical duplicate is that, and then so there are basically what happens in linear sequencing. It's hard to separate between optical duplicates and PCR duplicates. Okay, so there's only blanket term which we call PCR duplicates. Some of those reads come from the sequencing platform, what Mr. just explained, and the other part comes from the library frame. So together, whatever we are what we are seeing is actually coming.
Yeah, so the tool can even somehow split between what considers an optical or not. We just put it together and we go.
So, and each library type, there are some callable limits, like for the variant calling, when we are doing whole genome analysis, usually we make PCR-free library, and the number what we are looking for is around 10-ish. If it is more than that, and we don't expect it to be more than 20%, 20 is high, but if something around 10%, we like it.
So today I'm saying a lot of so. I mentioned the base quality scores recalibration. So the idea comes from if we observe a variant in the population that we know it existed in somewhere else, most probably if we see in our sample, it may be real too, and not an artifact. So that's the global idea, right? So what it does is, so we have a known set of variants that we can use. It's going to look about those mismatches. If it's present on the reference, we're going to assume it's real. And if it's not present, I'm going to recalculate the quality scores, take into account more things. The position in the read, whatever's around it, and taking the original quality scores also in consideration to calculate a new quality score. So we're going to be modifying whatever the aligner tells us about the quality score of the base to something different through this process, by taking into account more information. Can you see a problem here? Or a problem, or you think the problem is going to be whatever we use as a reference. It's not necessarily correct, right? Sure, but I mean, I'll give you an example of probably that it's not necessarily correct. So one of those is dbSNP. So all these IDs are from dbSNP. So dbSNP also has errors, I'm sure. It's not going to be a big deal, but they exist, so you need to be aware. The realignment, reassembly idea, so here I was trying to give an example of before and after realignment. So here is the problem, right? Before realignment, you're going to detect two SNPs that already are present on a database, so you assume they're real. But when you do the local realignment for that indel, you realize that they're gone, right? So are these reals or artifacts that somebody else published as correct when they are not part of they actually don't exist and it's a problem of the alignment?
But in the second one on the right,
Yep.
there is a deletion of 4 meters, like here.
So this would be like
the other RSID on that. No, there's 3, 4, yeah.
Oh yeah. So this represents the indel, yeah.
So that was already there on that also.
Oh yeah, it's present but not called to some extent, I'm assuming, because there's no support, or was called probably, yeah.
So it improved after you did the
local realignment. But again, this is not happening. So local realignment looks, so the alignment failed because basically it takes every read at the time and it doesn't have information about the rest of reads. Local realignment looks, okay, there's a situation here. Can I improve it somehow? Are these correctly aligned or not? And it comes with the conclusion that if probably this is more real than this local situation. again, no, it's not happening anymore in JTK4. It uses a different approach. But I thought,
Why did they decide to do away with this?
I'm assuming they consider the new approach better. I don't know the details. But I mean, we've been using JTK3 for a long time, right? During the teens is what we use the most. I don't know when we started to use JTK4, but we've done a lot of analysis following these steps.
So that, but the previous slide is just showing the known set of variants. The T was the known set of variants.
Yep, all were known.
Okay.
All were known. So they passed the previous filter of base quality recalibration, but not the local realignment.
So what we are seeing in DBSNV, some of them may actually be false calls.
That's what I'm assuming, yeah. Again, I'm not an expert on those databases to tell you how many are wrong or not.
So are they correcting it when they have been proved experimentally? No, this is not a
I don't know. I have no clue.
because we rely so heavily on that.
Or maybe they exist, and this example was just not there, right? But because it existed in the database, we call it, yeah, this is fine. We see it in the database. Maybe they were real, but not in this sample, definitely not in this sample.
And these wrong calls were picked up on the IgG. After looking them on IgG, they were, I mean,
Well, there were examples of this situation more than anything else. We don't go into that deep of the detail, right? We generally, I mean, not in production settings like we are, right? We trust whatever the aligner do and whatever the JTK tell us, and we worry about other things more than going back into the detail. Here I'm pushing or giving the idea or giving examples of what happens in the background.
So are
You are not going to see all that, by the way.
commercial pipelines like Dragon also using JTK or they have got different algorithms?
They change, right? I mean, the first idea was to use JTK. Yeah. So they use something, their own color right now. I don't know, I haven't, Bonnie, do you know what color Dragon uses? They have their own, right?
Yeah, they have their own. They are actually driving servers actually, and they are GPU accelerated, so they have their own product.
So a Dragon server, what it is instead of having a computer with the software is inside of the chip itself. So that's why it's so fast. They have a name for those chips. I forgot how they call them. No, they have a specific name. PG, I don't know. It's been used in other settings, not only. So Dragon chip is not new. It's been used for other things. So the last steps, basically, if you have multiple samples, we have a specific calls for every sample independently, but you can do another thing. So especially if it's a big cohort, you can use that information of the cohort to improve your calls. Because you're expecting that cohort is going to be more or less similar. So you're expecting similar calls. So you can even improve your final set of calls using the cohort information. If you're working with less than, I don't remember, 50, there's some sort of cutoff that we generally say that we consider that for good for joint calling, cohorts, or just individual calling. In any case, if you you have multiple samples and you call them separately, what you want is to put them in a single file. So that's generally what we do. Even if you have 3 samples, you're going to end up with a single final call after some filtering that we can do. And we will do some filtering.
This final BCM merging, this is only for cohorts or like you do it for?
No, we do it even with 3 samples.
So it's for a trio also you will have a final BCM?
Yeah, with a single one, yeah. We generally do that. I mean, you can keep them separated if you want, but it's simpler to work with a single file.
So but it will have information, for example, if you're working with a trio.
Yep.
So in the same BCM, you will have variants of the father, mother, and the child.
Yep.
In one BCM, and
Yep.
mark which is coming from there.
Yes, so, and we'll see it in the practice. There's a little bit of a complication for that, but yes, that's how we keep it.
But how do you know which was coming from where?
You give the genotype. So there's going to be columns for genotype for the mother, father, and proband, for example.
Based on HLA or based on what?
No, the genotype of that position. So if you see a variant in one of the 3, you call that, it's going to be present in the final file, and then you're going to say it is...
So you sort by positions then?
Yeah, your final list is going to be sorted by chromosome position, and you're going to have the genotype of every individual at the end for that specific position. Oh yeah, so the next thing, so the BCF is our final product, is variant calling file. That's the name we give it, BCFs. You can have them like BAM files. We have some BAM. You can have them compressed, and then we call them BCF with a B. I'm really bad differentiating between the two, but yes, V and B, BCF files are the type. There are specific tools to work with it. There are some columns, the minimum number of columns, basically chromosome positions, the reference allele, the alternative allele, the quality of that call, if it passes filters or not, there's information about that. And the info field. Info field can be really big. With like also exists in the BAM, we can have a lot of information about the alignment, and it can change depending on the variant calling or what you do with that file. But yeah, we can have a very big info field too, with information about the variant. So this is how it can may look like for a simple sample. We're going to have a bunch of header lines, but after that, every line represents a call or a variant. Chromosome position. You can even have ID information there, like DB SNP IDs, if you do that or annotate them somehow. Reference, alternative base. The quality scores we've been mentioning all the time. So you notice that it's a different scale from sequencing that goes up to 40, alignment goes up to 60 normally with the things we do. Even you can get 90 for a call. If it passes another filter, the info field that I mentioned can be really big. Here I'm just giving a quick example. So this is the depth of sequencing, and this is the allele frequency that you observe. So what we're saying, how many times you see it? You see it 56% of the time you see the alternative. And the format for the sample that is going to come later. You can have multiple samples here. This is the genotype. The number of times you see, in this case, is the genotype. It means, are you familiar with this annotation? Being 00 or being 01 or 11? No? Okay, sorry. So I need to probably stop here. I think I have. Yes, I do have what it means. So we generally use 0 for the reference. So if we have, so we have two copies, right? So of the information. So if we say 00, it means it's homozygous reference. If we say 01, it's heterozygous. If we say 11, it's homozygous alternative. If you are working with a lot of samples, you can be in situations that you have even two present there, right? Because there's some sample that may have a third allele that is not present in the others. It can happen, I guess. Especially if you work with indels, and the indels are weird.
So in the sample 1 is heterozygous. So what is the other?
Yes, so this is reference allele, the number of times you see the reference allele, the number of times you see the alternative. So I'm assuming these should give you the 56 if I didn't made up the numbers, which I'm not sure, because this is just one thing that I put here. I probably put some numbers on, but I'm assuming if you do this division, it may give you the 56, I guess.
What about genes where there are copy number variations?
Well, that's a different problem. The assumption here, or maybe the first pass analysis, is that we did two copies, right? But yes.
Some genes which might have as many as 6 copies, 8 copies. So how do you do that?
Well, that's you need to use something different, right? Or estimate the number of copies in a different way. But for variant calling, the caller is going to assume that you only have two copies. Yes, we do it. We do it with exam data. And generally, the copy number situation that we're talking about now is generally solved based on depth of sequencing. It is the basic information you have, or the only information that you have.
No, but then you'll have to have a mean of a number.
Yeah. Yes. But there are callers for copy number variation even in exon. So there are specific tools for copy number variation in exon sequencing. They generally use the concept of that idea, or the basic concept behind it is based on depth. Right? You expect that all exons should have the same depth for sequencing, and suddenly one or a whole gene changes the depth of sequence. You're giving a probability of having a copy number situation. That is really common when we work with somatic calling. When you work with tumors are really unstable, and copy number variation is something that you need to analyze when you work with tumors. 14 is the number of times that you see G. 18 is the number of times that you see A. So there are 14 reads that align in this, or there are a total of 32, right? 14 plus 18 aligning. 14C and a G, 18 they see A on that situation. Sorry, my throat is itchy now.
That is 32.
32 is the depth, again, is repeated here. It's telling you based on this. And 99, again, quality, genotype quality. How confident are we this is the right call? All this information I'm explaining, I don't know if you're going to see it in the hands-on, but generally it's in the header. It's explained. The header are really long, telling you all the information that I'm just telling now verbally about your file. Even myself, there's so many codes for these things, sometimes I get confused. I don't know them all on top of my head. Oh, here, I did it. So this is a header. I wasn't sure if I include this. So if you're curious about the DP, the depth of sequencing, it tells you approximate rate of read depth, and GAQ, genotype quality, and so on. You have pretty much a lot of fields that you can have there. Probabilities in different type of approaches, you can have them too. We also talk about this, Dr. Amit talked about this. Yeah, we have our calls, but we don't know what they do. So we need more information. So we're going to add annotation to it. There are different types. There are different tools. Here I'm mentioning 3. So this is the 3 we used in our pipelines. We even use more than one, which is for really specific things, which doesn't make much sense, but we do.
But the outputs would be different.
Yeah, but we have to merge them, which is an extra work we do in it. But I mean, we build a pipeline, but we merge them, yeah. For specific reasons, but we're going to use today VEP. It was a pain to make it work in our system, really a pain to make a VEP to work. That's one of the reasons, now I'm going to get a bit technical when we talk about content environments, that what I did here is not ideal, which is I created a content environment for every project we've done. From bulk RNA-seq to single cell to variant calling is a single gigantic environment. Don't do that. Divide it by your projects or type of analysis you're going to do. Because then you encounter that the variant effect predictor doesn't work. Or it was installing a super old version to make it compatible with the rest. It worked for a day and it crashed after that and it couldn't make it work. So the way again, I'm going to get technical, but just to give you an idea of solutions, if you need to work with solutions, to make it work, we use a container. You know the Docker containers we talk about? So this is in a container by itself inside of our content environment to make it work. It wasn't another way because we couldn't make it work to make it compatible with all the other tools that were available. So again, so for annotation types, we do a lot of different things. I'm mentioning here a few, but one of the most important ones is what is the consequence? What's the impact of this, right? It is a loss of function call. Loss of functions, I think I probably mentioned later. So let's go through the next slide so I explain what type of annotations we can get. And the main goal, right, or one of the main goals, especially in variant systems, but probably in anything or a lot of type of analysis you want to do, the reason of annotating is because you want to do some cascade of filterings, right, depending on your project. But generally you want to do some cascade of filterings to explain your phenotype, whatever phenotype is the one that you're studying. Yeah, we can start with, I think on average, I think mentioned we have a lot, between 3 and 5 million variants among us. Not all of them are very important. You can classify them based on the consequences and say, of course, a synonymous variant is not going to be important for my setting. I'm going to remove those, and so on, and so on, and so on, and so on, until you probably get one or more, depending on your setting again. But if you're working with a rare variant, the expectation is this one explains your phenotype. So that's what you're trying to get. So the impact, we were talking classification of the variants based on the impact. And I talk about loss of function, and I realized I maybe didn't explain what loss of function means. So VEP classify them in high, moderate, low, and modifier. And these are the types of loss of functions that I was trying to explain. If you find a stop codon in the middle of an exon, you know that protein is not going to be functional. Or it's going to be highly not, most probably it's not going to be functional. maybe functionally if you're lucky and it's the last exon of your protein and it doesn't do anything important and it's probably something that, okay, you can have situations maybe that the protein survives somehow with a stop codon maybe at the very end, right? But yeah, you have other things, like if you have an indel that changes the frame shift, you know that it's going to have a high impact consequence, right? One of the biggest problems, missense variant, you know there's a change of an amino acid. That's going to be hard to figure out if that change of amino acid does anything. For that we use other type of annotations, but they could be interesting, definitely. And the low impact. We don't know what a synonymous variant may, well, we're going to assume generally that a synonymous variant does nothing, but they may do something. And we even have cases there are cases, for example, that modify another variant, right? So,
What if they are at the splice site?
well, yeah, if it's a splice site, we can even, I think, yeah. So a splice donor variant.
I mean, a synonymous variant which is lying close to the splice site.
Yep.
It might change the.
I'm sure. So there's never like a. So you can never be completely sure that it's not doing anything, right? We talk about the missense variant. So there are tools for that they try to make an effort of classify them as pathogenic or likely pathogenic or not pathogenic. Sorry, likely pathogenic, likely. They can. I shouldn't use that word, but it depends on different. They have different names for everything. We can say it's a deleterious variant or a tolerated variant, a benign variant, however you want to call it. So there are multiple tools, and you are not going to be safe only using one. That's probably the message that I want to say, is that one is not going to solve all your problems. There are discordances between them. Some of them are based, like meta-SVM, meta means some sort of meta-analysis. You're going to put together more than one of those estimates and try to give you an answer. Others are based on, they're based on different systems. I'm not going to go all through them, but the main message is that one is not is enough to give you an answer. And generally we use, I think in the current pipeline we use 7, and the one that we use. 7? I think it's probably some number around those numbers. That's the one that we generally use for missense variants. For intronic variants, we use another type, but also they exist for intronic variants that try to give you an estimate of the probability of that variant affecting the protein. I don't know, the popularity of those, if I need to choose one, we work a lot between CAT-scores and meta-SVM and REBEL. So REBEL, meta-scores, and meta-SVM, they were the 3 that we use a lot at the end of the CMG project. We're the ones that we trust the most. But I mean, you cannot trust a single one.
REBEL is also a meta tool. I mean, it combines.
Yep. Another important one, right? For rare variants, I think you heard that before. The expectation that a disease is being caused by a common variant is probably not. It's unlikely, right? Especially if you work with rare variants, you expect that the variant you're studying is going to be less than the prevalence of the disease, right? Significantly less, but because a lot of diseases are not explained by just a single variant. There's going to be multiple variants that explain the disease, so for sure, your variant needs to be less than the prevalence of the disease. Assuming full penetrance. The penetrance is a different problem too, right? Then we have ways to annotate for frequencies, and there are different databases for that. Being Nomad, a popular one, and you have it for genomes and exomes, and they're being combined in a single one now. Conservation, that's another way you can annotate. If your variant is being conserved in a lot of a species, you know, or the amino acid is conserved in a lot of a species, you know that amino acid, if it changes, it may be an important change that you need to study. If If it's an amino acid that changes in a different species, you can think, okay, yeah, I mean, it happened before, it may not be. If it happens now, it may not tell us much about it. There's measurements about intolerance. So if you're working with a gene that has, in the normal population, has a lot of variance, you can assume, okay, a few more, it may not change. It may tolerate variance compared with another gene. So that's another thing you can look at it. There are different databases that already include variants and can give you information about it, like Cosmic, I think we talk about it, which is for cancer. These two are for clinical variances. OMIM is not for variants, but it's for genes. How many people, I don't know, I don't know how many people here is a clinician and uses these things, but this is for variants and this is for disease. OMIM, are you familiar with OMIM? No, it's for Mendelian. Yeah, it's a popular. Oh, okay. OMIM, yep. Cosmic, I said Cosmic is for cancer. So there's a lot of databases that you can.
ClinVar and OMIM, a lot of clinicians use now.
Yeah, totally. The problem of annotation tools is always you need to update them because those databases they update. So the tool you use need to be updated too and so on. It's really hard to keep track. This wasn't a slide that Bonnie showed. And I want to show it for a different reason. What year was 2011? Whole genome sequence of siblings. I don't think they were twins. And this was the process of filtering, right? You start with 6 billion bases, and you keep going the cascade of filtering up to you get, here's 3 candidate genes, and there was only one that was shared between the siblings, and both of them were affected. So they decided, okay, this is the most likely gene that causes the phenotype.
What was the disease?
I don't even remember. But it was a coding variant. That I remember. So could have done whole exome sequence and solved the same problem? Yes. Yeah, the last slides, I think I did only a few more. Let me know if you have questions. The problem of calling somatic calls is different than calling germlines. We use a specific callers, which is part of JATK, that is called mutic. Oh, there are more. This is not the only one. You have other options.
But the somatic variant can also occur at different times. It could be just post-zygotic, it could be much later.
Yep. So here we are kind of representing the evolution of a tumor, and you have a problem of clonality also in the tumors, right? So if you have your analysis, you do your analysis here in time, and you can imagine, I'm going to come this in time, I have the tumor, I'm going to do the analysis at this time point, you're going to observe, and you may have different clones in there, meaning different clones having different variants. So now the frequency that you see a variant changes, and the number of variants that you can have, they can change too. So this is a hard problem to solve. So tumor clonality is the main issues, right? Germline callers are not going to work with this. The copy number variation that we mentioned earlier is also a big problem. Purity is a problem too. I don't know how many times pathologist comes or you send samples and they tell us, or somebody that works at the Department of Pathology and tells me, oh, this is really pure. And then when we do the analysis, it's not, right? And try to explain that it's not. There must be cells in your tumor that are normal. And the data tells us that it's not pure.
There are some cells in tumors, immune cells.
Yeah, of course. They're infiltration, right? Yep. Oh, here I'm just trying to explain the problem. But yeah, you got the problem that purity and all the factors we explained earlier, it's always going to be a problem. And as such, you need to go deeper in sequencing.
So for tumors, you do an exome with a high coverage, or you do a panel?
Oh, in the clinical setting, they may use panels for research. I don't know. I'm not familiar with clinical settings so much. I don't know what they do, but I'm assuming they can do both, probably.
No, I'm asking that for tumors because you need high coverage to get the...
Oh, well, they do. We modify the whole exome pipeline, for example. We spike in to cover 500 genes in a higher coverage.
Those are sequenced at 600x coverage coming out. So those 500 genes are sequenced at 600x coverage.
So the rest of the genes you will do at
150x coverage.
and then the others you will do at the higher
500x, 600x.
So how do you come to know that, I mean, the data is from, not from tumor, but from the normal, I mean, the surrounding cells?
Oh, we use frequency. We look across all the heterozygous calls to calculate that. So, you know, based on expectations that 50 and 50. So based on the frequency of the alternative allele across the whole data set. So in theory, if you should expect at least 20 in an ideal world, right? So a pure 100% pure tumor, the head call should be 50%. Any deviation from that may be caused, of course, by biases on the sequencing, but also by not being pure. And you can analyze all the sets, and you could assume that on average, you're going to get a good estimate of the purity. We observe purities between 40 and 60, and that's totally normal. Below 30%, it can be problematic. You're going to be missing a lot of calls. Again, it depends on the, all depending on the coverage, right? We have the problem of the coverage here. But let's say in a standard setting, we generally say, again, I'm not a person that works a lot on tumors to tell the best approaches for this. I would say, I think what we 40 and 60 on purity, totally normal. Below 30% starts to. So about long, why we use applications dealing with the structural variant. Whole exome sequences for these things, right? We are not going to structural variants with whole exome sequence. If you and you short reads, they are short, and ideally you want to cover at least the breaking points, or go over the lead or over the structural variants to solve it, because it's going to be, it's a complicated problem if you work with short reads. Tandem repeats, of course, another problem. We can use them for phasing. That's if you care about phasing, right? So you can imagine that in the same gene you have two calls, and they are far apart from each other. You don't know if it comes from the same allele or from different alleles. If both of them are stop codons, I'm making this up, or they are loss of function calls, if they come from the same allele, you may think, okay, that's fine because you have an allele that produces a protein that is functional. But if it's in a compound heterozygous situation, that's how we call it, then you have a problem because they come from different alleles. A way to solve these situations of phasing those calls is with long reads, because you can cover both of them and you know if they come from the same allele or from different alleles. Of course, full transcript sequencing is another application. And probably there are others, of course. Assembly of the genome. And that's it. And my favorite, I wanted to finish with my favorite quote. So yeah, talk to your analyst if you are not the analyst. It's always good. Designing the experiment in advance is always good. Okay. Oh, we still have an hour before lunch. Should I start? Yeah. So if you have questions, that's a good time. I need 2 or 3 minutes.